So hi, we're from Mr. Khanna's lab, and today we're going to be presenting a comparative genomic analysis of colorectal cancer microbiome bacteria to discover novel relationships. Okay, and so colorectal cancer is uncontrolled tumor growth that starts in the rectum or colon, and there's many factors such as daily habits, genetics, and the environment that may impact the development of colorectal cancer. But usually what is often ignored is the gut microbiota that plays a role in the onset of it. So our research primarily revolves around the gut microbiome, which is the community of microorganisms living in the intestinal tract that are vital to human health. And there have also been more recent studies that have shown a positive correlation towards colorectal cancer. So we want to study that. So we specifically are targeting bacteria in the gut microbiome, and our specific research purpose is to target pathways impacted by shared proteins of both probiotic and antibiotic bacteria in order to decrease the malignity of gut tumors by increasing the expression of inhibitors of colorectal cancer. So our research is mainly dedicated to helping the wet lab groups in their experiments to narrow down bacteria regarding the expression of colorectal cancer to help accelerate their experimental processes. And so our main end goal is to target and screen these unique pathways and enzymes of the cancer-associated and non-cancerous associated bacteria to decrease the metastasis of colorectal cancer tumors. And the analysis of bacteria present from a recent cancer microbiome review paper is also being conducted to reveal novel phenotypic and genotypic differences at the protein and signaling/pathway levels. So our main ultimate goal is to analyze these differences in pathways and enzymes between cancerous and non-cancerous associated bacteria in the gut microbiome. And we hope that by narrowing down these bacteria associated with the non-cancerous status in the gut, we can help find more efficient treatments for colorectal cancer. Okay, and so the main computational tools that we have used so far include BV-BRC, which is the Bacterial and Viral Bioinformatics Resource Center, RStudio plus R programming, and NCBI Blast. So we initially used BV-BRC, which is an information system based on research on viral and bacterial infectious diseases. And we mainly use this platform to run comparative analysis tests of our target bacteria, and also to create figures such as phylogenetic trees, pie charts, and multiple sequence alignment graphs. We also use the programming language R, which is mostly used for statistical computing and graphs, in conjunction with RStudio, which is an IDE, in order to create tables that compare our cancerous and non-cancerous data and genomes. And then initially, we also utilize NCBI Blast in the beginning of our research, and this is a platform that helps us find similar regions between biological sequences through comparing nucleotide and protein sequences. And so our team specifically used this to compare our non-cancerous and cancerous genomes in the initial stages of our research.
So the very first thing we did was use BV-BRC to create two groups of genomes of the bacteria, which separated the cancer-associated state bacteria and the non-cancerous associated state bacteria. After this, we used a bacterial genome tree test to make a phylogenetic tree showing bacterial relatedness between the two groups and inside the two groups. After that, we ran a comparative systems test on the genome groups, which made a list of EC numbers. EC numbers is just a classification of proteins on what their function is. Because of this, a given bacteria can have more than one of the same EC number. After that, we used RStudio to find two EC numbers exclusive to cancer-associated genomes and two exclusive to non-cancer-associated genomes. We then ran a multiple sequence alignment test, or an MSA, at the genome level, comparing each of the EC numbers' respective genes across the genomes. The MSA also yielded a position versus entropy score graph, which helped us pinpoint areas of similarities. After RStudio, we were able to find two tables. Each of the tables showed a list of EC numbers that are exclusive to each group. For the non-cancerous associated bacteria, deoxyguanosine and deoxyadenosine kinases were exclusively found in the 4 out of the 10 non-cancerous associated bacteria, and each of these are linked to purine metabolism. For cancer-associated bacteria, we found lysine 2,3 amino and a phosphatase, which are linked to lysine degradation and liposaccharide biosynthesis, respectively. And these were exclusively found in 13 out of 23 cancer-associated genomes. The very first figure we got from BV-BRC was a phylogenetic tree. This one was comparing all the bacteria in each of the groups, and this can help us find two bacteria that are related to each other based on the nodes. If the nodes are more connected, therefore they are more related. If the nodes are spread apart, therefore they are less related. This can help us with further testing to compare areas of similarities.
Now, going on to the multiple sequence alignment, this graph is the graph we got from after running the MSA for the protein lysine 2,3 aminoamutase. So lysine 2,3 aminoamutase is one of the two proteins that are exclusive to the cancerous-associated genomes. And then the other graph we got while running the MSA was this entropy versus position versus entropy score graph. And this is the graph of the protein with the EC number of 3.1.3.82. It has a really long name, but the short form for it will be GMHB. GMHB is exclusive to 13. It shows up in 13 out of 23 cancer-associated genomes. And with our previous knowledge in chemistry, we inferred that the lower the average entropy score, the higher the level of similarity. And the reason we showed this graph, especially for GMHB, was because GMHB had the lowest average entropy score, and so we could infer that it had the highest level of similarity among its whole genome. Future steps, we created figures which included pie charts, phylogenetic trees, MSA models, and pathway maps. And these figures displayed the results of cancerous and non-cancerous associated proteins and its pathways. We did it through BV-BRC. And through this research, we attempted to reveal the novel phenotypic and genotypic differences at the protein and signaling level. In the future, we aim to target and screen the unique pathways and enzymes. And as of right now, we are currently analyzing the mRNA sequences of the 4 proteins we found, and also the pathway these 4 proteins are part of. In the later future, our final goal is to create a probiotic drink against colorectal cancer. We would like to thank our advisor, Mr. Clinton Kunha, for guiding us through our research project. And we'd also like to thank ASDRP and the Olive Children's Foundation for providing us the opportunity to conduct this research.